Good morning. I'm Mark Rohrbaugh from the NIH Office of Technology Transfer within the Department of Health and Human Services. It's my pleasure to moderate today this panel with extensive experience in technical considerations to discuss the role of new and existing technologies in adjuvants and technology transfer models. So without further ado, I'll introduce the first speaker, Dr. Robin Robinson, who is the Director of BARDA within the Office of the Assistant Secretary for Preparedness and Response in the U.S. Department of Health and Human Services. Robin.
Thank you, Mark, and thank you for the opportunity to address this group today with a few remarks on the roles of existing technologies and adjuvants, meaning egg-based vaccines and adjuvants. And let's see. Yeah, this is a slide that I have used for probably 2.5 years. Some of the assumptions here are going to be debated probably in February at WHO, meaning the pre-pandemic vaccine stockpiles. But I draw your attention to two things. One is the recombinant vaccines and egg and cell-based. My colleague, Armen Donabedian, will talk to you more about cell and recombinant-based, and I want to address egg-based vaccines and adjuvants, which would go across not only egg and cell-based vaccines, but indeed recombinant-based or molecular technologies that could be used for vaccines. But the point of this slide is to show that with all of these different types of vaccines available, then one can have an impact. But you know that they have to be at certain points in the presence of a pandemic. Pre-pandemic vaccines for H1N1 were not available, so we lost that weapon as we went into this pandemic. Recombinant vaccines were only one was really far enough along that even could be considered on a worldwide basis. And so primarily what we were left with were egg-based vaccines with at least 2 small cell-based vaccine capacities available, and when those would be available. I draw your attention that recombinant vaccines, if they meet their promise, could be available within 12 weeks because of for the first doses, because of their way that the potency assays are done may be a little bit different. But cell and egg-based are going to be about 22 weeks. And in reality, we've always said about 20 to 24 weeks was the range, and it turned out with the H1N1 vaccine, it was about 21 to 24 weeks. So it was about as anticipated. Was there a great difference between the two? Not really. So with egg-based vaccines, let us remember a few things. That it does provide currently about 99% of the total amount of seasonal and certainly the pandemic H1N1 vaccine manufacturing capacity globally. And even regardless of you're an industrialized country and developing, the eggs right now are what we have available. The vaccine safety and effectiveness of these products has over 50 years of successful history here. And so that's not to be denied, and certainly it's an incumbent industry and everything that means. And there are very good things about that. It means that the system for making the virus reference strains are in place, the reagents that are necessary, the process of going through that, selecting those strains and actually going through eggs is well known, it's well understood. And it also offers the advantage of being able to rid the virus isolates of adventitious agents in many cases that other technologies would not have. Secondly, the vaccine manufacturing processes and facilities are in place. And in fact, they've been expanded and new ones have been built. The technology is well known. Again, the systems and the processes are well developed and well understood. Doesn't mean they're not vulnerable, but they are well understood. The potency assays and immunogenicity assays for the product and for clinical studies and preclinical studies are well known. They may be not as fast as we had hoped, but they are well known and well understood. And finally, and I think probably most important we've seen with the H1N1, is that these are licensed vaccines and they are well accepted. In the U.S., about 30% of the population receives the seasonal influenza vaccine, and in some cases in polls we've heard that as many as 50% of the people wouldn't want an H1N1 vaccine in the U.S. So I don't think that's to be discounted at all. I mean, these are vaccine manufacturing processes that are well understood. They the products are very safe, and they've been shown to be very effective. But these bulk manufacturing facilities are very specialized for being able to pick those eggs up, to inoculate those eggs, to cut off the top of the egg, and to remove the allantoic fluid with the virus. And you can't use it for anything else unless you have an egg-based vaccine. So at least the upstream manufacturing process is very specialized, which then leads to certain other things. There are vulnerabilities with egg-based vaccines that have been mitigated over the years by having very careful quality control systems. And one of those is the exposure to avian pathogens. Egg farms that are used in the U.S. for making these vaccines are well controlled, and they are shown to have very low levels of pathogens, and there are redundant systems to obviate any type of problems with avian pathogens. And the last time in this country we had a major problem was in the 1980s. The egg supply. If you can't get the eggs, then you can't make the vaccine. And certainly one of the things that in the U.S. we took hold of, the very first thing was that in 2004 you had to secure a year-round egg supply. If you're going to make a vaccine for not only seasonal influenza vaccine for the Northern Hemisphere in the U.S., which meant that you started making vaccine in January and you went all the way into June and July, that you also had the capability to make a pandemic vaccine, which could be any time of the year, or even a pre-pandemic vaccine, which would be in the off-season. So that's the other part of the year, it's usually the fall. So you had to have a year-round egg supply, and secondly, that egg supply had to be secure. It had to have redundancy, a contingency flocks that could come online and actually have those birds that were producing the very best eggs all the time, every day. We did that in the United States with the first bit of pandemic influenza vaccine manufacturing money we had, and that has gone forward and has been able to double the amount of vaccine supply we've had in the U.S. Secondly, it took about a year and a half to do, and we will be expanding that as we go forth. The next thing is that the virus strain growth is important and is a vulnerability for egg-based. But it's also a vulnerability for cell-based vaccines. Until we have other vaccines that do not depend on the ability of the virus to grow, we will be susceptible and vulnerable as we were this year with the H1N1 pandemic. And in fact, it was about and some estimates, twofold to fourfold drop in the supply that we had expected going forward from the late summer into the early fall because of that one factor. And Dr. Donabedian will talk more about how the other types of technologies may be able to obviate that. My final point here, though, is that this incumbent industry, which has served the world very well, will coexist with newer vaccine technologies, and at least in the lifetime of most of the people here, will be probably the primary means of making influenza vaccines. So I tell you this and say this, that we should not just discard them immediately and throw them to the side, because they still have a place for a long time. The next remark I have is about adjuvants. We have seen aluminum hydroxide provide adjuvant effects with many different types of bacterial and viral vaccines. In fact, there are even licensed influenza vaccines that are not used anymore that have aluminum hydroxide. Many studies have been done over the past 5 years looking at H5N1 viruses, with H1N1 and others, H7, H9s, with the effect of aluminum hydroxide as a dose-sparing measure. And overall, it's been very limited in the dose-sparing effect. Over the past 10 years, though, there have been oil and water adjuvants that have been developed primarily by several manufacturers that do demonstrate dose-sparing effects. In H5N1 vaccines, as much as 12 to 24-fold with that vaccine. With the H1N1, we see is anywhere from 2 to maybe 8-fold effects. Additionally, cross-reactive immunity among virus strains. As mentioned earlier, with these adjuvants, you could actually see the H5N1 virus vaccine become immunogenic against from one clade against another clade, and so that it does provide that type of cross-reactive immunity as another advantage. Thirdly, there are a number of studies have shown that it provides enhanced priming effects such that one could be immunized with a prime boost, with a prime vaccination, and come back as much as 5-6 years later with a booster and have a very high immunity effect. The next thing is there's limited licensure in wide populations. There is certainly a seasonal influenza vaccine that's used in Europe in the older population that has been around for almost 9 years and multiple millions of people. But there have not been millions of children that have been vaccinated to date with that. So there's the experience in all populations is somewhat limited. Secondly, the acceptance of the variable vaccine. What we saw in this country is it was hard enough to actually get people to accept this vaccine being licensed. And certainly if we had needed the vaccine to go forward, we certainly thought long and hard about it. But the acceptance is certainly one of the things that we have to consider going forward if we use adjuvants in either seasonal or pandemic situations. Certainly a pandemic situation with a very severe virus gives you a risk-benefit ratio that it may be certainly worth the benefit. So what have we seen from the H1N1 pandemic that has allowed us to say more about the adjuvants? One is that they certainly in the clinic, and certainly they have been accepted from a safety point of view, and certainly we've seen usage in certain parts of the world with this vaccine. Again, acceptance, whether it's because it was not seen as a severe pandemic or was because the vaccine contained adjuvants, I don't think we understand that yet. Lastly, there will be IP issues as we go from multinational companies to developing countries. And so those will be issues that will have to be resolved as we go forward. There are other adjuvants. There are other adjuvants that are being developed under the support of WHO, certainly those that are being supported with NIH and HHS funds. But the point I want to leave you with is that this is the course adjustment that we have to think in terms for pandemic vaccination going forward when these products have been accepted and then licensed here in the U.S. And that in order to have manufacturers be able to meet their seasonal influenza vaccine demand, they have to have facilities that can address that. That's much smaller than the amount of pandemic vaccine that we will need. The one way that we can actually have a 2, 4, 8-fold greater surge capacity is by the use of adjuvants. So I think that as we do our lessons learned and after-action results of the H1N1 pandemic, one of the things that we need to really look hard at is the effectiveness of these vaccines with the adjuvants. And I think then the data will probably come back and say that they are very effective, and that we will then hopefully move forward not only in the U.S. but all over the world with licensure of these products, at least in terms of a pandemic usage, but also there are probably niche or boutique markets for these products. Thank you.
Thank you, Robin. The next speaker is Dr. Armen Donabedian, who is the Senior Program Manager within the Division of Influenza and Emerging Diseases at BARDA.
Thank you, everyone. My offering to the workshop today is to provide a BARDA perspective on the role of cell-based and recombinant flu vaccines in pandemic preparedness. I think this slide deserves a closer look because it summarizes our influenza pandemic preparedness strategy, which is aimed at weakening rather than stopping a pandemic wave sufficiently to prevent the impact of the pandemic from outstripping existing healthcare capacity, which is shown by the solid gray line at the bottom of the figure. The pandemic wave is shown in pink, and as you can see, the use of appropriate countermeasures can reduce that demand by depressing and spreading and delaying the pandemic peak. At the same time, by supplying, increasing the capacity of essential countermeasures such as ventilators, oxygen, antivirals, etc., to increase the healthcare capacity, it is possible to get the most out of our healthcare system and thereby ameliorate to the greatest extent possible a pandemic. Now, if we are fortunate and we can anticipate what the strain will be, it is possible to use pre-pandemic vaccines to blunt that pandemic peak, perhaps in combination with adjuvants. However, if the strain is entirely novel, then we have to rely on our existing capacity to make flu vaccine. As Dr. Robinson pointed out, that would be both egg and cell vaccines. However, as he also pointed out, the difficulty with that approach is this will typically take 20 or 22 weeks and may well not be available. Vaccine may not be first available until after the pandemic peak has passed. Now, to meet the need of earlier vaccine availability, to address that gap between the possibility of pre-pandemic vaccines and standard vaccines, BARDA has made is strongly supporting the advanced development of recombinant influenza vaccines wherever feasible and within our mandate to support technologies which have demonstrated feasibility, that is to say, have reached phase 1 clinical trials and look safe and immunogenic. Preclinical studies and the preclinical status of other technologies, especially work being done at the NIH, will be covered by my colleagues. Now, a major priority for BARDA has been an effort to capitalize on the advantages offered by cell-based flu vaccines, starting with the incentives, providing the incentives to transition from egg-based to the more robust cell-based influenza vaccine manufacturing technologies, and supporting work towards licensure in the United States. So as you can see from these pictures, we'd like to go from chickens and eggs to cells and bioreactors. Now, there are many potential advantages of cell-based influenza vaccines over egg-based vaccines. They are better controlled. There's less risk of contamination. They're more expandable on short notice. There's no need to create or maintain a long supply chain of flocks or wait for new flocks to be brought online. It avoids constraints and risks of the egg supply. What I mean by constraints is that some vaccines, for example, live influenza vaccines, may require specialized eggs, pathogen-free eggs, which may be difficult to obtain all over the world or may be very expensive. Cells would bypass that constraint. Also, there are risks in the egg supply. Of course, H5N1 may not grow well in eggs. In fact, may be lethal unless we are fortunate enough to make a reassortant, which will allow effective growth in the eggs. Cell-based vaccines will also avoid egg allergies. Importantly, They may also allow the isolation of better matched vaccine strains. And we have heard this point brought up many times, and I'm eagerly awaiting data from the CDC and others to see whether, in fact, this is the case. It's commonly understood now that many relevant H3N2 strains cannot be isolated in eggs and therefore cannot be considered vaccine candidates. And this could be a severe problem. If a properly matched strain cannot be isolated in eggs, we would face a disaster. Cells offer a much better chance of isolating the best vaccine candidate. That means there's less chance of a vaccine mismatch, there's less chance of a delay while we try to find a strain that will grow in eggs, and it will prevent vaccine shortages brought on by those two earlier issues. The majority of H3N2 strains, greater than 65%, can be isolated in cell culture. In addition, viruses made in cells may have an improved immunological profile over egg-derived vaccines simply because they're grown in mammalian cell lines, although data is pending. Finally, the manufacturing platform used for cell-based vaccines can be used to support next-generation vaccines in so much as the cell substrates could be used in the same bioreactors. So these considerations caused us to provide awards. We made a major effort in this area, and we've awarded 6 contracts worth $1.3 billion in 2005 and 2006 to support the advanced development of cell-based vaccines. Advanced development means development beyond phase 1 clinical trials towards U.S. licensure. And in return, we have requested a commitment from our contractors to develop a plan and to work towards developing a surge capacity of 150 million doses within 6 months of a pandemic onset. We awarded 6 contracts to Novartis, Baxter, Sanofi Pasteur, GSK, Solvay, and MedImmune. The status is that 2 of these manufacturers are completing phase 3 clinical studies, and they're expected to submit BLA shortly. 2 have struggled and remain in early stage development, and 2 manufacturers were down selected mainly because they did not want to make the commitment to build manufacturing facilities to meet surge capacity requirements. So I think that's a pretty good scorecard. It's a very difficult undertaking, and 2 successes and 2 still in the game is about where we thought we would be. One company has been sufficiently successful to receive a follow-on infrastructure building contract. This company is Novartis, and in January 2009, we awarded a cost-sharing contract to this company totaling $486 million to design and construct a U.S. capacity with a production facility with a production surge capacity of at least 150 million doses of pandemic influenza or some other emerging infectious disease. In addition, the contract the contractor must provide at least 2 commercial scale doses lots each year for up to 20 years. In November 2009, the facility in Holly Springs, North Carolina, opened for MF59 adjuvant production. This facility is available for emergency vaccine production, will be by 2011, and we expect that they'll be licensed for cell-based influenza virus manufacturing in 2012 or 2013. And I have some pictures that show the beginning of this in 2007. Some of this construction was supported in the cell-based facility contract the cell-based process development contract, which I mentioned at the outset, where we paid for some of their equipment. So that company began construction in 2007, and you can see the progress through 2008. And this is what the facility looks like now, the cell-based facility in Holly Springs, North Carolina. A symbol of a strong government industry partnership resulting in the first U.S.-based cell-based vaccine. Now, there are a number of significant challenges when making a cell-based vaccine. First, manufacturers are concerned about overcapacity. They're concerned about the viability of the business case. They don't see why they would want to make a major investment when the improvements may only be evolutionary rather than revolutionary. For example, they may not meet the gap that we talked about that recombinant vaccines can meet. For live flu vaccines, which may incidentally have a cost advantage since the cost of Spophus eggs is so high, cells may provide a cost advantage. The problem that we have there is that there isn't an accelerated approval process for that vaccine. It requires efficacy studies. There isn't an immunogenicity bridge that's currently accepted. And this complicates the development and increases the cost very significantly. Also, FDA's concerns persist about the safety of vaccines produced in mammalian cells, particularly a live flu vaccine where you don't add an inactivating agent. And so anything that may be cryptic in the cell line may emerge and cause an unforeseen problem. Comparison of egg and cell-derived vaccines are complicated by the fact that vaccine seed is derived from eggs, and so one-to-one comparisons may not look favorable for cell-based vaccines, and it's unclear in many instances whether they are equally more or less immunogenic. Finally, the value of cell-based investment versus the promise of the recombinant technologies is a problem. And there are some, it is thought in some quarters that it would be best to essentially skip it and go to recombinant vaccine. But the truth is, cell-based vaccine is a proven technology. And BARDA needed to move forward with a technology that we were relatively certain would result in improvement in our pandemic surge capacity. Back to this again. Those recombinant vaccines, because they can address, the promise of those vaccines is great because they address that gap in between pre-pandemic and cell-based vaccines and standard egg-based vaccines. As we've said now repeatedly, it may provide vaccine sooner. And we were able to identify one company who met our criteria for advanced development. And we awarded a contract to that company in 2009 for $155 million to begin advanced development of that vaccine. And in return, we requested that they develop a surge capacity of 50 million doses that could be provided in 6 months of a pandemic onset. That company is Protein Sciences. They use a purified HA protein from baculovirus. They are currently completing phase 3 clinical studies, and they are expected to resubmit their BLA in 2010. In addition, we have recently issued another RFP, and we are expecting to provide additional awards to recombinant and molecular technologies for influenza vaccines in 2010. To wrap up, the challenges with recombinant is this is a new approach, a new technology. There are only a few companies that have demonstrated feasibility in phase 1. And so the risk is very high. Also, the companies that are producing these products, developing these products, they are smaller companies, and they do not have the resources, either monetary or technologically, to move quickly. And they require a lot of support from BARDA. So the discussion question that I'd like to leave with you, and hopefully we can address throughout the rest of these meetings, is how are these technologies translatable to the developing world? And finally, contributor and now acknowledgements. Thank you.
Thank you, Armen. Our next speaker is Dr. Douglas Holtzman. He is a senior program officer at the Bill and Melinda Gates Foundation.
Thanks very much. I'd like to thank the organizers for the opportunity to participate today. at the stakeholders workshop. It's a privilege to be here, excuse me, to present on behalf of the Bill and Melinda Gates Foundation. I had the flu and I've got a persistent cough. I'm going to... Thanks. And so my talk is going to be a little broader than the last two talks, and my goal is to share the key programs that we're supporting that relate to the sustainable influenza vaccine capacity. That's the key interest of this presentation. And I also want to address the question of why we're engaged in the area. It's a question that I get asked quite often because for the most part the Gates Foundation focuses on neglected tropical diseases. And I'm asked not just by people outside the foundation but also by my colleagues in the malaria team and TB team. We supported, along with the U.S. government and the Wellcome Trust, a meeting in Beijing about 2 years ago. I don't know if you know Dr. Jeremy Farrar, who heads the Welcome Unit in Vietnam. Some of you may know him. And he gave his presentation and started off saying that H5, which has obviously been a big driver for all the technologies that we're going to talk about, that more people had died of lightning strikes than H5 over the 10-year period that had generated all this interest. And so I guess my point is in all of this is I wouldn't want to be quoted as saying that I was happy that there was an H1N1 pandemic, but I think it does justify and point to the fact that all the preparation and all the work that we've been doing is really well justified, needs to be supported, needs to be continued. So again, to kind of sketch the bigger picture of the Gates Foundation's engagement, as I said, our involvement started in about 2004. We had a briefing by Dr. Keiji Fukuda and some of his colleagues talking about the H5 situation and the impact that the H5 pandemic virus might have on global health. And as many of you may know, the core mission of the Gates Foundation, or the core belief, is that all lives have equal value. And so with respect to pandemic influenza, really equity is the key driver for our involvement. I mean, we know even before Chris Murray's article in The Lancet and the modeling that's been done that it was pretty obvious that the burden of disease was likely to fall disproportionately on those in developing countries based on lack of access to vaccines, lack of healthcare systems, et cetera. But I'm the strategic program team lead for the pneumonia effort at the Gates Foundation, and influenza is a pathogen within the pneumonia program. And so for us, it's really not just about pandemic influenza. We're really involved in flu for much broader reasons because as the CDC and others and much of the work has been really showing recently, influenza is an underappreciated burden in the developing world. And I think I'll touch on that, and perhaps we'll get back to that in some of the other sessions. And so our projects really have been targeting a couple of key areas. One is to follow on again from the funding of the U.S. government and others, looking at the burden of disease and using vaccines as probes. Also data for decision making in general, and then I'll finish my talk with focus on the new technologies, which are really mostly preclinical. So one area that we've supported is the area of maternal immunization. So this is work by Dr. Mark Steinhoff, who was at Hopkins but was moved to Cincinnati. And in the interest of full disclosure, the maternal immunization program really started with a focus on pneumococcal vaccines and safety issues around pneumococcal vaccines, but it wasn't an accident that flu was chosen as the control arm. And what that study showed, and I think what the H1N1 pandemic that we've just experienced has really shown, is the sensitivity of pregnant women to influenza. And so this strategy of using maternal immunization, as I said, we're focused on flu right now, but it's really meant to be a way to reduce the burden of disease in the developing world by immunizing mothers and having them transfer passively protection to their infants. And so I don't know if we have, does one of these work as a, I'm afraid to push the button here and let's see. Oh wow, look at that. In any event, I'll just leave that aside and say that, as you can see, when the mother has a high degree of antibodies, these will get transferred transplacentally, and then following on from that, following on from that, the immunization in the child could lead to protection. So the result of this study, though, was pretty surprising, which showed a huge burden of influenza and a marked reduction in the mothers who'd been immunized. So we're following up very actively on this. We're also very delighted to be able to support Abdullah Brooks in following on some vaccine probe studies in the infant population following the epidemiological work that's been supported by HHS, which again shows that a large burden of severe pneumonia seems to be associated with influenza. So the next program I wanted to share with you is really probably best suited for the lab session tomorrow, but I figured I would share it in advance. And, I'm calling this pre-surveillance. I mean, one of the things it seems to me is that even with all of the cooperation that occurred and the best surveillance activities that one can do, which are essential, obviously, that there was an opportunity to sort of try and think ahead of the virus, if you will, that there was some, reason to believe that one could explore the molecular space of the virus in the laboratory to try and identify sequences and viruses that might have pandemic potential and therefore might be viruses of interest, shall we say. And I don't want to get too much into the molecular biology here, but some of you will be familiar. There's been studies by Ab Osterhaus and Dr. Yoshikawa Oka showing that the affinity for the receptors in the respiratory system are different between the avian viruses and the human viruses, and it's an absolute assumption in this program that the human receptor, this alpha-2-6 story of the binding in the human upper respiratory tract, it would be a component or a likely component of a human pandemic virus and allow transmission that drives this program. So in brief, what Dr. Kawaoka, who's the principal investigator on this program, is doing is taking avian viruses, using mutagenesis techniques and high-throughput technologies to generate billions of variants in the laboratory, to use affinity approaches to sort of capture the viruses that seem to be binding to human receptors, and then to study and analyze those, to sequence them, to assess pathogenicity, to do antigenic cartography, and to generate the antibody reagents that would allow one to explore the antigenic relationship between those viruses. And why is this important? Why am I even telling you this? I mean, we saw a 3 to 4, 5 month delay, really, in the availability of the reagents to even analyze the vaccine. So even if the recombinant approaches could deliver antigen in 2 months, which I believe they can. I'm a technology optimist, acknowledging all the challenges that these products are going to face. I think we still need to try and think again ahead of the virus to try and pre-position reagents, if we can, qualified viruses, antibody reagents, etc., that would allow us to respond much more rapidly to the outbreaks that we see in the field, in surveillance, in humans, in animals, etc. Okay, so let's go on sort of more for the data for decision making. And so we've supported work at PATH in partnership with WHO under contract by Oliver Wyman. I'm not going to go through this in great detail, but we really felt there was a lot of work that needed to be done on supply and demand and the logistics of the stockpile. As you'll recall, before H1N1, really a lot of the discussion was around how are we going to prepare for H5, and there was discussion of pre-positioning vaccines in stockpiles so that would allow an immediate release of vaccine against a pandemic that was anticipated to have a much greater case fatality rate. And we've worked closely with WHO and other partners to try and bring as much data to the table as possible. And in addition, during those studies, one of the outputs of that was really a technology landscape. I think that Laszlo said that some of those materials that WHO has done are in the book, but PATH did its own technology assessment. And breaking it down very simply, we've got the vaccines we have today. We've got the ones that are a bit intermediate in terms of some of the adjuvants, and we've heard those questions. A nice presentation by Robin and Armen. And then there's the sort of the future state of the recombinant vaccines. And even beyond that, to these sort of broadly cross-protective vaccines that everybody would like to see develop, but are 15 years away at the earliest, and may never be achievable given the biology that we're facing. But in and amongst that set of opportunities, really we've gone after some of the recombinant approaches. I mean, I think The presentation that Armen just made, I think, should be a little sobering. I mean, I think it is a success for the U.S. government, and I think that the cell-based vaccines are an important intermediate step. But, how many countries out there can really afford to invest, that kind of resources into cell-based manufacturing? And to answer the question that was raised by the woman from the World Bank, whose name I don't know, we do have an assessment really of some of the comparative cost analyses for the different types of vaccines. And in the end, it really, some of that information is on the PATH website, and you can speak with the PATH representatives here. Some of that is available publicly, but the bottom line is that the cell-based vaccines, the inactivated cell-based vaccines, I should say, to be specific, are unlikely to come in at a cost of goods that would probably allow them to be used on a regular basis in the developing world. And here I'm talking about the public sector in the developing world. Given the financing challenges, even for general vaccines that are, childhood vaccination, it seems unlikely that sort of seasonal programs are likely to be created in the developing world in any time in the near future. And again, that's part of the reason, that's part of the challenge between the supply and demand that we're all facing here. So let me just tell you about the specific programs that we have been supporting. The major program is at PATH, and I'm not going to spend that much time talking about it. John Boslego will be speaking about this. But they're supporting 3 different groups right now. The Institute for Experimental Medicine to develop some additional pandemic vaccines in parallel with the H1N1 work that's being supported by WHO. A small company called Lentigen, which uses lentiviral technology to generate VLPs, as well as New Genesis, which is an even smaller company using Neurospora crassa, a fungal system to try and generate large quantities of antigen in virus-like particle format. Before the initiation of the PATH program, we had supported 2 separate programs. One was at the University of Texas Medical Branch, and it was to support a program that they had in collaboration with a company called Vaccinate. Some of you will be familiar with this program. It's an M2E base, so it's another one. It's this common protein approach, and by fusing M2E to flagellin, it was hoped that this would generate a robust immune response. M2E is not itself normally highly immunogenic. That program completed its phase 1 study, and we're no longer funding that, but the program is continuing to try and resolve some of the dosing issues and associated with the initial material, and they have a very active program still. And then the last program is a plant-based technology, a transient plant-based technology that we're supporting at the Fraunhofer Center for Molecular Biotechnology. That program is also getting support now from DARPA, the Defense Advanced Research Program, which is another part of the U.S. government. And for us, this was really, again, I'll touch on sort of the why in just a minute, but this program is really the primary focus of the technology, or we think the primary candidate is really malaria vaccines and malaria transmission blocking vaccines. But I think when you think about the challenge of how do you make the business case, or how do you, as we talked about, how do you create a warm base for some of these technologies, there's sort of 2 options I see. One is that we use a lot more seasonal influenza vaccine, or the other is that you have dual-use technologies that you can trade off. You have trained people, active facilities, a warm base, but that could be switched over to pandemic vaccine manufacturing. And but again, these programs are at least certainly the plant technology is very, very high risk at this point. It's all preclinical, but we are hoping that alongside of DARPA that it will move into the clinic in 2010 or perhaps early 2011. And again, the logic again behind this, I won't spend so much time on this, but coming out of the PATH assessment as well as our own assessment was that for the developing world, we really needed, both for cost issues and the pandemic response, we needed technologies that could be very inexpensive and really scalable. And so that leads to the set of technologies here. And again, in terms of the strategic focus, it's really about innovation for the foundation. We were not. We understand the expense associated with some of the near-term technologies, and we're not investing in physical plant per se. But we thought that if we could help de-risk some of these technologies, make them available through sort of global access strategies with interested partners down the road, that this would be a useful contribution that the foundation could make. And so in summary, the Foundation's involved in a range of activities. Again, I didn't have a chance to mention the etiology work we're doing in childhood pneumonia, but alongside the vaccine probe studies, we are interested in sort of further defining what is it that's killing children of pneumonia. It's the number one killer of children, and often, 20-30% of the cases, it's unclear what's the cause. So influenza will be part of that. It'll be a multi-site and multi-pathogen study. I talked about the vaccine probe studies as well as the risk assessment, the sort of pre-surveillance project. And then, so alongside the supply-demand work from Oliver Wyman and the investments in the technologies, I think I've kind of shared with you in very rapidly the program that we've developed in support of pandemic and seasonal influenza. So thank you very much for your attention, and I look forward to the discussion.
Thank you, Doug. Our next speaker is Dr. Jan Hendriks. He is an account manager for international support at the Netherlands Vaccine Institute.
Thank you. The Netherlands Vaccine Institute is a national institute under the Ministry of Health in my country. Our unique values used to be and still are over 100 years of know-how of vaccinology in the public domain. We are independent from industry, and we have a sizable and highly developed production infrastructure covering the entire value chain of vaccine production. Last year in February, the Dutch minister decided to start a process to sell the manufacturing part of the institute towards industry. This process is still continuing and is going to be completed by 2012. This means that there are some challenges regarding how to maintain for public international activities our capacity, how to make it remain accessibility for partners in developing countries. And this process is still going on. But what is sure is that the research and development capacity, which is now in our institute, will remain accessible, because probably this part will be reunited with the National Institute of Public Health, which is on the same premises of our institute. Over the years, the Institute has had a large tradition of tech transfer, not so much on IP generating and then licensing to developing countries, but more in transferring know-how of, say, public domain, EPI, traditional vaccines. A notable example of this is the World Bank vaccine project that was executed between 1990 and 1998 with Chinese partners, 3 partners, on DTP, measles and OPV. We will not talk about this project in this short presentation. An interesting precursor of our current project with WHO on flu was our HIP project, which is still ongoing. We redeveloped HIP conjugate vaccine technology to transfer it. That was the specific aim and the purpose of that project, to at least 3 to 4 partners in developing countries, where we do not put an exclusive license but a non-exclusive license, and negotiated reasonable fees for in some cases a royalty arrangement to recoup some of the investments of this product that was not developed for our own market but for tech transfer purposes. So I spent most of the time of this presentation on our collaboration with the WHO, with what we call the ITPIF project, the International Technology Platform for Influenza Vaccines, that is based on egg-based production approach. And this project and this approach of tech transfer actually is different from the HIP project approach, as that is not going to negotiate or charge royalties with prospective partners, because basically most of the project is funded by international public money. More recently, we also began a project of collaboration for tech transfer on a new polio vaccine, which is considered to be more safe if it is produced in developing countries. The same in IPV project. Our hip conjugate vaccine development and tech transfer collaboration is regarded by all a number of standards as successful. We developed a pilot process in our institute that was based on public available knowledge. The John Robbins technology was used, and we have transferred this to 4 partners. And the most notable success was that in 2007 the Serum Institute obtained a license, and I think, to my knowledge, is now having an installed capacity of over 100 million doses of hip conjugate vaccine. The Saban project was, as I said, started more recently, and in this slide I just wanted to show you that we use our public technology available and developed in our institute over the years on viral cells grown on microcarriers for Salk IPV production. We will use that approach for redeveloping this for strains on the Saban strains. And the parts of this project that has a threefold subdivision: clinical lot preparation, proof of principle studies, clinical studies, and training and tech transfer. The bold parts of this overview are funded by a grant of the Bill Melinda Gates Foundation to the World Health Organization's Polio Eradication Initiative. Then the International Technology Platform for Influenza Vaccines. In fact, the concept was suggested to us by WHO, the so-called hub concept. Laszlo has already indicated our involvement in this in previous presentation. The idea was to develop very simple egg-based production process that is already used for many, many years with good yields in order to be able to transfer this by training to interested parties. So the idea is not so much to generate IP, but to make available all the know-how, the hands-on know-how which is needed to actually start up production in another institute. The selected technology was the inactivated whole variant influenza vaccine, as I said, in eggs. I'm sure nobody can read this slide, but you can maybe still see the colors. And the colors indicate the different elements of our project. In blue, on the left upper side, this is all the activities we had to do to install production capacity. NVI was not a manufacturer of influenza when the request came from WHO, so we had to procure and install, validate, commission, etc. The equipment for the pilot scale production. In yellow, we show here the production process as it was developed. In green, the assays, the QCAs, and in orange, which happens to be the color of our queen, our nation, the tech transfer activities are indicated. This scale of the pilot process is 10,000 eggs, and as indicated in previous speakers, it's another set of work if you start working with eggs, if you are used to cell culture, as we do usually in our institute for polio, for example. You go back in terms of dimensions of waste that you have to handle, so we had to invest in a lot of things that we were not used to anymore. Use semi-automated equipment, and we have a process. We have chosen a very simple basic process based on a seasonal strain, monovalent, in order to mimic and to prepare for pandemic strain production for later activities. The current timeline where we now is that we have completed a number of initial parts of the project, and we have also started on training and technology transfer agreements. As of next year, we will also endeavor into more sophisticated process development aspects, as I will mention a few minutes later. The process is not, as I said already, is not new. It's based on zonal centrifugation to purify the allantoic fluid which has been produced in eggs previously. And I show this slide just to show you that in the training component of our project, we try to visualize the process of purification in the zonal centrifuge of the viruses by asking the trainees, can you, if you understand the process, can you show the right order of the different steps of how the virus behaves during the purification? We have also, of course, had to meet criteria in terms of purity, and we show this slide as just a proof that we can have a rather pure product using our setup. In terms of yields, there's a kind of generic minimum requirement that you need to have for the 10,000 X scale, but also for large scale. You should have at least one, but preferably 1.5 or even more doses per egg. And this overview shows you that we manage in our latest runs to meet that requirement. In terms of activities on this tech transfer part of the entire project, we did 4 main things over 2009. We established the website, which is meant as an instrument also to keep up in touch with trainees that come to us afterward for follow-up exchange of information and support. We run a QA GMP workshop for 13 participants from 7 countries. Most of them were representatives from the WHO grantees, as has been explained earlier to you by the WHO presentations. And we have started a first generic training course for 10 participants from these WHO grantees countries. And I'll show you some more information on that later. And in addition, we have searched for partners, and we are still continuing that will have engaged into more permanent bilateral tech transfer agreements. The training course in 2009, we have just completed that, was for us an enormous amount of preparation because, as I said, we had not done this kind of processes before in the Institute. And since it's a hands-on course, a bench course, and even where we show a manufacturing process by making a training demonstration bulk production, it requires an enormous amount of capacity and motivation and commitment from our staff and our facilities. So we are very proud that we managed to do this successfully. Of course, in the evaluation, apart from all the nice words that the trainees told us, there's also a lot of criticism, not criticism, a lot of suggestions to improve. So we will have to redo and improve for our next editions for 2010. In order to give the best generic and overview of capacities needed, we decided to make the group in 2 subgroups, one for production aspects and one for QC. And also we had some generic lectures in advance. That also is a challenge to organizational limitations, but I think we also selected and we asked institutions, because eventually here we don't try to do individual training, but we try to do institutional capacity building. So we asked beforehand, institutions who are interested, please come to us, but give us 2 people, one from production, one from QC. So we have 2 different programs in this 1 3-week course. The access that we do provide is basically very technical and very detailed, maybe for most of you here, but it's on technical advice, a lot of facility design, utilities, they are the key before starting manufacturing, process and production technology, how to make seed lots, how to get the zonal centrifuges to work. An extensive amount of documentation was presented and provided on a USB stick, and as I mentioned, we have a 2-volume course manual that gives the backgrounds and all the details, including worksheets. We invested in a number of activities for assays, in process control training, release tests, assay validation, qualification. We spent a lot of time in discussing with the participants how to calculate, how to interpret results. It was seen as a major difficulty by some of the participants how to actually calculate data and put them into perspective of what you want to achieve. And also we have spent time on preclinical support in terms of how to develop a clinical plan, etc. So a lot of lectures were given, but the practical things were appreciated most by the participants in the evaluation. Then finally, in addition to this generic training that we will repeat at least twice in this year, we have managed to start an initial consultancy agreement with Vaxera that will last about for 3 years, where we will provide advice and consultancy where Vaxera needs in a larger framework of their project that has been mentioned earlier. With Vietnam, we have in December agreed a similar arrangement, although at present a bit more modest in terms of funding that was available and in terms of time. But as indicated, the PATH group is going to work extensively in Vietnam, and we hope to be part of that engagement as well. In Iran, we are anticipating a fact-finding visit somewhere this year to see if and how NVI could additionally contribute to their capacity increase. And we also noted there is a lot of interest now from other countries, from China, Kazakhstan, Argentina, India, and South Korea. We have received applications for our 2010 editions of our courses. So I think we are well on track, and the planning for 2010 is to confirm the process as we have established now in our hands, to develop this process further into subunit vaccines to meet the legitimate concern on sustainability, in the sense that if you have a whole virus vaccine, that's never going to be a marketable product in many countries for seasonal. And since the project was designed for pandemic, we focused first on the pandemic whole virus approach. But most partners will also need a subunit market, and therefore we will also extend our process with a subunit step. And then, as I mentioned, we will do at least 2 training courses in March and April. And we have to agree to carry out the agreements just signed with Egypt and Vietnam, which also include, among consultancy and on the spot training, include 2 tailor-made group training activities in our institute. To finalize, I think I kept in my time because I still see a 2 minutes sheet here. A few pictures of the course. This is the group that came to us in December with some trainers. The eggs, as we also had to learn how to handle this, including the waste. The trainees, it's also a challenge in a GMP environment to work with participants that want to see and do everything. So here we see the harvest of the eggs, and the zonal centrifuge was also keenly watched by the participants. This was the production part. Separate, we did this QC training. Here we see our participants working on this damned SRID test, which is for regulatory purposes and needs, but it's a challenge for reproducibility in many ways. We also forced the students to speak up and present their own institute and present their challenges. So we tried also in seminars to address challenges that go sometimes beyond technical challenges. This is Dr. Bae, the new director of the IVAC Institute in Vietnam, explaining his facility. And then most of the work was done by my colleagues in the institute, but we also are proud to have a kind of international faculty. Professor Osterhaus gave a very exciting and comprehensive, as he used to do, overview of the whole world of vaccines. Erik de Hond acted as the international production expert. Laszlo Polkoniai was teaching the students how to handle the eggs. But the most important external visitor was not this one, he's the second to most important. That's John Wood, overseeing the QC training. This was the most important, Santa Claus, who happened to be in the country when the courses were. Thank you.
Thank you, Jan. We have about 15 minutes for comments or questions, if anyone has any. Dr. Bloom?
Barry Bloom. I had to give a lecture on flu, and I called it, "Why Are We Making a 50-Year-Old Vaccine?" And before giving it, I went on the web, and I read the 200 first citations on Google that came up if you typed in H1N1 vaccine. There was only one citation at that time that had anything positive to say about any vaccine, and the remaining 199 were the most scurrilous, distorted, untrue allegations about vaccines that absolutely shocked me. So in my talk, I understood why I would have been fired if I had been able to have a modern vaccine released. But the question is, we've talked today about supply and new supply. We haven't talked about demand. And so the question is, how do you deal with a public that is subject to unbelievable amounts of misinformation, and yet we're trying to get seasonal flu vaccine as a habit or a commitment in order to prepare for a pandemic? What's the U.S. government or any other government doing?
Okay. Thank you, Barry, for the question. Certainly one of the things that we had to deal with during the H1N1 pandemic has been the myths and misinformation about influenza, about vaccines in general, and specifically about influenza vaccines. And I think we've probably done a very good job with that in taking them one at a time as they've come out and not letting them sit and fester. That is something that I will applaud my colleagues at CDC of undertaking and doing a very diligent job of going forward with that. With seasonal influenza vaccines, will we be in a situation where we will have an adult immunization program where that's where we have everyone in the country, at least in the U.S., recommended for that? I can't say that is going to happen this coming spring, but if you look over the past 4 or 5 years, you've seen that the number of people that are recommended for seasonal influenza has increased actually dramatically as we've gone forward. The manufacturers will tell you there is a sweet spot or a threshold in which they can manufacture and still be profitable going forward. We're not there yet. And when I look at the numbers, I actually see that we actually grow in the United States like adolescents. We don't grow like this. We go like that in increments. Certainly, this is a good point to plug that this is a national flu immunization program week, and that the CDC and the HHS will be going out this week promoting. It's still enough time to be vaccinated for H1N1, and don't forget about seasonal influenza also. So I think those are some of the things that are challenges for us in public health to go forward with, and to continue to be vigilant about that.
Is this on? I just wanted to follow up. I don't know if anyone saw the New York Times article about a week ago, because it had a very, very nice joke by Dr. Offit in it, and I don't know if this will translate for the international audience, but you're right, we're schizophrenic about this, aren't we? I mean, you have half the people saying they're pounding the table, how come I'm not getting my vaccine? On the net it was 99-1, and then a large number saying this is unsafe, I wouldn't use it, I won't give it to my kids. And his joke was, two people sitting at a restaurant, and one says to the other, the food at this restaurant is terrible. And the other one says, I know, in such small portions. I mean, it's something we've got to grapple with, and it's really, it's a big challenge.
Daniel Miller, if I could address some of the answers to that. I think if you look internationally, with the H1N1 experience, we have a lot of lessons that we need to review. Because across the world we see uptake rates in the developed countries ranging from 6.5% to 75%. Most importantly, I think that there is a lot of applied research that can be used here, because even in H1N1 initial polling data, for example, and surveys, and this is an observation you see with any preventive service, that one of the best predictors of uptake of preventive services such as vaccination or mammography or pap smears or other things is the recommendation of people's personal physician to have it done. And that's an area that CDC has been working quite closely with, but I think all of us have a long way to go.
If I can add something on that, this is also something that we have really noticed at the World Health Organization. It really seems that the way the medical studies are organized in many, many countries is that there is virtually very little or nothing about preventive medicine using vaccines. So these physicians, in the private practice, the general practitioner, they don't know about vaccine. If they do know something, they know something which is 20 years old or something, and there's no refresher course. There's no way these guys would be able to give the correct information. So they themselves, they look at the websites, it's full of anti-vaccine lobbies, and this is what they advise their patients. In the Geneva area, some of my colleagues say, you know, I don't even dare going to a visit to the GP, because every time they know I'm working at WHO, I get insulted. So, so I think that there's really something that the governors could maybe do and try to incorporate more knowledge and more teaching about prevention, about vaccination, about immunology in the medical studies.
Yeah, I have a follow-up question. This is Derek Carter from IDRI to Barry's question, and I'm wondering is, in the context of this workshop where we're talking about new technologies, whether the feeling is that public acceptance of new technologies would be better than old technologies, because a lot of the propaganda is directed against existing vaccines. So for example, we're moving a little bit away from squalene because of the propaganda against squalene to develop a new oil-based adjuvant. Is the feeling of the panel that that would be a better thing than the old, or trying to prove the safety of the old?
Again, I think this comes back to what Barry asked in his question, and that is myths and misinformation, and that it is incumbent upon us to provide the scientific and medical facts for these and not let them fester, not let them stay there, but to give that, and we have to do it over and over and over again. But it will be a challenge as we go forward to re-educate not only industrialized populations but in the developing world the benefits of vaccine. And I think Tony Fauci always points out when he gives his vaccine talks, you know, we have been able to save hundreds of millions of lives through vaccination over the past 200 years. And when you put it into that kind of perspective, I think that you know, we have to use that as one of our tools. And again, just to be upfront, transparent, and explain, you know, what the facts are in a way that people can understand them, and just to set the data straight on it.
Sorry, I don't necessarily want to leave that area, but I wanted to get back to the question that Dr. Donabedian left with, and see if he has an answer, or maybe better. Doug from Gates, and the question is, you know, is there a possibility that we could have a platform technology to rapidly produce a vaccine that could be taken into a developed country and set up in a sort of a modular fashion? Is this something that Gates is interested in, or, you know, with sort of cookie cutter training and the whole aspect of technology transfer, that it's realistic? Or are we going to have to start from scratch in developing countries, training, developing at the most elemental level to do the technology transfer for those companies or countries that don't have any sort of technology? I guess my question to either one of you.
I'm happy to share my opinion, but I may not be even the best person to speak to this. There's others who have been sort of doing the heavy lifting of the tech transfer, and those who are out in the audience who are trying to receive these technologies. I guess I am a technology enthusiast, but I don't want to short sell the huge challenges of the staff and the GMP mentality and the culture of precision that really has to accompany vaccine development. I think most everybody in this room understands that, and God forbid that we have a couple of bad severe adverse events or a bad lot of flu vaccine. You can imagine it would set us back 50 years. I mean, so I think it's a difficult question to answer. I think for the foundation, there really is a long-term commitment to prevention. We believe deeply in the power of vaccines, and so the investments that we're making, I think, are entirely rational, but they're also very long-term. And I appreciate deeply the tension that many of the folks here on the stage probably had to grapple with as they thought about, don't just stand there, do something. I mean, we're in the midst of a if H5 goes, it's going to be a catastrophe. We've got to start moving the ball. And how do you trade off the sort of near-term certainty against long-term risk? And it's a difficult equation. I can't give you a simple answer, but I do believe that if we pick the right technologies and they are given the right resources and the partners are interested in it, that, I would like to believe that, 15 years from now, maybe that, eggs would be on the wane and the new technologies would be getting put into place and more actively deployed, because I do think they would benefit all of us, but that's supposition.
You have time for one final question, comment?
I would like to comment on the safety of the product, this new H1N1 vaccine in Egypt. Actually, we started to receive this vaccine from GSK last November, and we ordered for 5 million doses. And in fact, by time we realized that part of the people, not most of them, but part of them, they are against using the vaccine because of the testing finalization. Because what was said in Egypt by the Minister of Health himself, that there will be some sort of liability on ourselves. It's not only on the company. So the doubts starts to come to the people how we are going to use a vaccine that is not well tested or not yet approved. So can anybody tell me when will be the deadline for this debate to get rid of it? I mean, what is the time to say that this vaccine is 100% safe, or at least to reach the safety of the normal vaccines? Because even myself, I don't have the answer. Thank you.
Well, as far as we know and everything, I don't know if David what to add on that, we are in regular conversation with the regulatory authorities throughout the world. We have no reason to think that the GSK adjuvanted vaccine or any adjuvanted vaccine is any less safe than the classical non-adjuvanted vaccine. Those of you who have received GSK vaccine may know that it hurts a little bit more in the arm, maybe. But apart from that, this vaccine has proven very safe. So what we are aware of that in different countries there are different worries. But we should also maybe try to be, in terms of us in public health and vaccine, try to have a consistent message. Yes. For example, a country close to my heart has suddenly announced that now finally they have got non-adjuvanted vaccine and finally they can go and immunize pregnant women and children. This is disastrous for the other countries who have only one vaccine, which happens to be the adjuvanted vaccine, and who have to use it in all their population, including the pregnant women. So I think all these noises about unsafety of certain vaccines are fueled by this kind of negative comments given with very good intention. So as much as we can know, and again, David may feel free to comment, there is no safety concerns with this vaccine, and 200 million doses total of pandemic vaccine have been distributed and used.
Well, let's thank does someone have a comment? Okay, let's thank our panelists once again for interesting presentations today.
Lunch is now available across the hallway in the solarium.